All right, hello and welcome to this last but not least session. Before we begin, let me mention a few housekeeping items. If you have questions for the speaker, please enter them into the Q&A tab. Be sure to mention what this question is all about, and you have the ability to upvote questions for the speaker to answer. And if you would like to ask your questions live, please use the raise hand feature and I will bring you on the stage. And there are a variety of emojis available to give feedback to the speaker as well. And if for some reason you have to leave early, the video of this session will be available in a couple of hours after the session has completed. And now let me introduce you to our speaker. Our speaker will be Edward Zhao from Fred Hutchinson, and I'll let him introduce his talk and let him speak. So please, Edward.
Okay. Hi, everyone. My name is Edward. I am a PhD student in biostatistics at the University of Washington in Seattle. And so the work that I'm presenting today is it's work being done at the Fred Hutchinson Cancer Research Institute with Dr. Raphael Gottardo and Matt Stone. And so I'm going to go ahead and share my screen now. Okay, so hopefully everyone can see this. Okay, so in my talk today, I'll be demoing our Bioconductor package BayesSpace. But before that, I'll first give a brief overview on the spatial transcriptomics technology, as well as just some of the key steps in the analysis pipeline for spatial transcriptomics. And then we'll end with a demonstration of applying BayesSpace to some real data sets. Oh, and I'll put into the chat here actually some relevant links to our website, our GitHub, and also our Bioconductor page. So that's in the conversations chats area. Okay, so I think some of you have probably seen this analogy before, comparing gene expression profiling or RNA sequencing to analogy with fruit. So in this analogy, we start with bulk RNA sequencing, where we're only able to characterize the expression profiles of mixtures of cells. Eventually, the development that comes out is single-cell RNA sequencing. And here we're able to identify, or we're able to characterize the gene expression profiles of individual cells. So the drawback to single-cell RNA sequencing at this point is that we still aren't able to spatially map the gene expression, because the process of single-cell RNA sequencing involves dissociating the biological tissue. So there was a need for a new technology that both, characterizes gene expression at the whole transcriptomic level, while also preserving the morphological context. And so that technology came in 2016, and the name is just Spatial Transcriptomics. And so the idea here is that we are preserving the structure of the, in this case, it's a fruit pie, but in the more biological context, it's we're preserving the morphology of the tissue. So we're able to map gene expression to a spatial location. Now, the difference between single-cell RNA sequencing and spatial transcriptomics, beyond the spatial aspect, is that current spatial transcriptomics technologies aren't actually at the single-cell level. So instead of this high-resolution fruit pie that you see here, what we actually end up getting is a little more like this. So it's a bit of a lower resolution image where each of your units of observation, here we're going to call them spots, contains maybe a couple dozen or a dozen or two cells rather than true single cell resolution. Despite this limitation, the utility of spatially resolved transcriptomics has already been recognized this past year in the Nature Methods Method of the Year. So we've already seen spatial transcriptomics have an impact in helping us better understand the biology of things ranging from the brain, the prefrontal cortex, to cancer tumor microenvironments such as in breast cancer and melanoma. At present, the leading platforms for spatial transcriptomics are Spatial Transcriptomics itself, which is the original technology. And this was acquired by 10x Genomics a few years ago, and I guess basically upgraded into the Visium platform, which is a little bit higher resolution and more robust capture. And so here I'm showing two images taken from 10x Genomics' product literature that give examples of what the data for spatial transcriptomics look like, as well as the types of analyses that it enables. So on the bottom here, we have a immunofluorescence stained image of the mouse brain, and then overlaid on top is the spatial transcriptomics data itself. In this case, it's plotting some continuous feature, maybe the expression of a single gene, or maybe the total UMI counts. And then on the right over here, I have an H&E stained image, and overlaid on top here, instead of a continuous feature, we have a discrete feature. So this is showing a clustering analysis where we try to cluster together spots that are more similar. So beyond those two examples, this is a more complete or more comprehensive analysis pipeline for spatial transcriptomics data. So we start from the raw data, and then along the middle axis here, a lot of these steps are actually shared with the analysis for single-cell RNA sequencing. But the spatial nature of the data brings some new challenges to these analyses, and also allows for a few extra analysis steps. For example, we're able to now spatially map our features as we showed on the previous slide here. And then I mentioned previously that the spatial transcriptomics technology is not truly single cell. So there is a motivation here to try to, get closer to single cell resolution. And two ways to do that. One of the ways is if you have paired single cell RNA-seq data, you can use a deconvolution algorithm similar to what you would do in bulk RNA sequencing. And in this case, you'll be able to estimate the proportions of the cell types in each of your spatial transcriptomic spots. And then another way is to try to enhance the resolution of your spatial data. And in this case, you can do it by taking into account some information from the spatial structure of the data, as well as potentially the image data, which we saw here, which we know is at high resolution. So how does BayesSpace fit into this? Our package BayesSpace allows us to do a number of these steps, from normalization all the way to clustering and the resolution enhancement. So the core features of our package are to do a clustering analysis as well as a resolution enhancement of spatial transcriptomics and Visium data. But we also wrap some other existing methods for normalization, feature selection, and dimension reduction to make it easier to conduct your analysis. And we also have a feature mapping plotting feature in our package. So there's still a couple of steps here that are not green. How did those fit in? So BayesSpace actually uses the single cell experiment data format, and that makes it really easy to integrate BayesSpace into any existing analysis for gene expression data. So by taking that into account, we're able to do these other steps very easily using BayesSpace output as well. Okay, so I'll give a brief introduction to the two data sets that I will be discussing today before going into the more interactive demo part of the presentation. So this first data set is the dorsolateral prefrontal cortex published by Maynard and others in Nature Neuroscience this past year. And this data set includes a total of 12 samples of the human dorsolateral prefrontal cortex. Here is an example of one of those 12 samples. So the original authors of this paper did a manual expert annotation of the layers of the prefrontal cortex from 1 to 6, as well as the white matter. And so these kind of represent the ground truth of the biological tissue here. And then on the right-hand side, this is the original author's attempt to recreate the ground truth using a data-driven approach. So in this case, by doing a clustering analysis that really didn't take into account the spatial nature of the data. So this is very similar to just reapplying some methods from single-cell RNA sequencing and seeing how good your clusters recapture the ground truth. And so what we see here is that it seems like layer 1 gets captured pretty well, but a lot of the middle layers don't seem to really show that banded structure, although we are able to see the white matter separate out from the gray matter. But there's, in general, just a lot of noise in this. And we're thinking that if we take into account more of the spatial nature of the data, we'll be able to get some better results. And so I'm skipping, of course, all of the technical details of how our package works, since this is mostly a code package demo rather than a full scientific talk. But in this case, I'm reproducing the images from the previous slide. So here again is the manual annotation. Louvain is a pretty popular clustering method that's often used in single-cell RNA sequencing. And again, this is similar to what the original authors saw, where there's no good layer structure and a lot of noise. And then once we use BayesSpace, which takes into account the spatial nature of the data, we're able to get a lot cleaner clusters as well as better layers that we can see in the plot here. And so this is just one sample out of the 12 samples total. If we look at the other samples, we see that BayesSpace actually performs pretty well consistently. So in this case, on the y-axis, I'm showing the adjusted RAND index, which is a measure of how well our clusters are corresponding with the ground truth. So a value of 0 would mean basically it's completely random. And then a value of 1 would be perfect correspondence between the cluster label and the ground truth. So we're aiming for a adjusted RAND index that's as high as possible. And so that we see here are a number of non-spatial clustering methods that are sometimes used in single-cell RNA sequencing analysis. And then Gottardo and ST-Learn are two relatively recently developed spatial clustering methods developed for Visium and other spatial technologies. And so we see that our method performs better than the others, but there is a lot of other, there are a lot of existing methods and even more clustering methods being developed. Basically every month these days we get a new method on bioRxiv. So it's definitely a pretty rapidly expanding field. Spatial transcriptomics is a nascent technology, and a lot of the analyses, the methods for these data are still in their infancy as well. And so the second data set that I want to talk about is a melanoma data set. So this is actually on the older spatial transcriptomics technology rather than the Visium technology. And so here we again have a manual annotation by a pathologist of the areas of tumor, lymphoid tissue, and the stroma. And then this is an example of a previous publication's non-spatial clustering analysis. And I want to point out that there's definitely some interesting stuff going on here where we see heterogeneity of the middle of the tumor compared to the periphery. But one thing that I want to point out is that we aren't able to identify a lot of the lymphoid tissue that's pretty close to the tumor, although the stuff that's far away from the tumor does seem to cluster out. So we're not really able to separate the tumor periphery with the lymphoid structure that's annotated. And when we use BayesSpace to increase the resolution of the data, we are able to recapture some of this lymphoid tissue that is pretty close to the tumor. So this was a pretty quick walkthrough of spatial transcriptomics as well as BayesSpace and how it fits into the whole analysis pipeline. But at this point, I'm going to jump into the package demo based on those two data sets I showed just now. Okay, so I posted earlier in the chat the website as well as source code for our package. But here is the link to our package. We have some installation instructions over here. Our package is in Bioconductor, so it should hopefully install pretty easily for users. And so the first vignette that I want to go through is the getting started vignette. So really the only package or only packages that we need to load here are ggplot and the BayesSpace package itself. So BayesSpace allows you to load data directly from your Space Ranger output that the Visium datasets comes in. So this is done using the read Visium function. So if you have your own data, then you can load that in using the read Visium function. But in this case, the dataset, the melanoma dataset that I showed a couple of slides ago, is actually already preloaded into the package. So we use instead this get RDS function to load in the data. So now I have this melanoma data as a single cell experiment format. And basically, this is what you would expect in any normal single cell experiment, except there's a few extra columns, namely, the spatial information, the row, and the column here. Okay. So, there's a pretty minimal amount of pre-processing that needs to be done for this data set, for spatial transcriptomics data usually, but we have a function that helps you get your data ready for analysis. So this is the spatial pre-process function. So I'm gonna just copy paste it here and let it run. But basically the spatial pre-process function, the first argument it takes is the single cell experiment. So it's your data. Then the platform, it's either the ST for spatial transcriptomics or Visium. So these are the two possible platforms that we currently support. And then just a couple of things like the number of principal components, the number of HVGs, and whether or not you want to log normalize your data. So these are just some of the pre-processing and dimension reduction steps that spatial pre-process does as a one-liner. Yeah, so I can show the help page for this here. So here are the options. We can choose either Visium or ST. In this case, the melanoma data is done on the ST platform. So moving beyond the pre-processing steps, now we're at the actual clustering step. So, generally, we don't know the true number of clusters. In this data set, we actually sort of do have an idea of how many clusters there are, or at least some idea of how many biologically distinct regions there are. So in this case, there's 3 annotated regions as well as some areas that aren't annotated. So maybe it makes sense to use a total number of 4 clusters, which I think might have been what the authors of this original publication did right here. So they clustered into 4 regions. But there's also a way to do this in a more data-driven way. So we have a function called QTune, which lets us estimate the number of clusters in your data set. So what this does is it performs a, I guess, just a quick clustering for the number of clusters that you choose. So in this case, we're looking at all values between 2 and 10. And then we want to see whether what the number of clusters that's best is. So this function generally works pretty quickly. We're only we're not training the model too much. It's just a pretty quick run for each of these numbers of clusters. And then we can plot our data using qplot, as we see here. And so in this case, we're basically looking for some sort of elbow in the data for the likelihood. And in this case, it seems like somewhere around 4 or maybe 8 is a reasonable choice for the total number of clusters. And so I'm sure some of you guys attended the talk by Daniela Witten earlier today, where the number of clusters, the true number of clusters, is actually a really hard problem to solve, and sometimes it's a bit of an ill-defined problem, since the number of clusters it might not be really the question that you want to ask. But in this case, we have a way to use the data, try to estimate what is a reasonable choice for the number of clusters. So the more clusters you choose, the higher, like the finer grained your clusters will end up looking. Now the main part of the clustering, the package, is to perform spatial clustering. So once you've chosen a number of clusters, you can go ahead and run the spatial cluster function. So I'll have that running right here. But basically, the important features here are the number of clusters, which is Q. We have some other, these can actually be skipped mostly, but a lot of these are the defaults. So the platform is the Spatial Transcriptomics platform. D here is the number of principal components we're using to do the clustering. And then in net method, we're using mclust, which is a pretty popular finite mixture model method for doing clustering analysis. And one important parameter I want to point out here is the gamma parameter. So right here, I'm setting gamma equal to 2. If we increase the value of gamma, then what happens is that there will be more smoothing across the data. And if we decrease the gamma, then there will be less smoothing. So when gamma equals to 0, then this essentially becomes a non-spatial clustering. It will give results that are very similar to mclust if you set gamma equal to 0. But in this case, here are the parameters I'm setting here. We generally suggest running at least 10,000 iterations. But in this case, just for the sake of time, we only use 1,000. Although you'll notice that this function actually runs very quickly, since this is actually a pretty small data set. So there's only 293 spots in this data set. Okay, so once the spatial clustering finishes, we're able to kind of preview what this will look like by looking at the call data to see where the data is stored. So once you have finished running spatial cluster, your call data will get two new columns, the cluster.init as well as the spatial.cluster. So cluster.init is what your mclust initialization is giving you, while spatial cluster here is the result from BayesSpace. Okay. And then once we have this, we're able to plot our data like this. In this case, we see that we have 4 clusters. Here is the tumor periphery, the tumor center, the stroma, as well as the lymphoid tissue. But again, we notice that we still miss a lot of the lymphoid tissue that is close to the tumor. And the idea is that perhaps the enhanced resolution analysis will help us with that. And so this next step that I'm going to go over is the enhanced resolution analysis. So I'm going to go ahead and run this over here. For this, we actually recommend running at least 100,000 iterations. So this will definitely be, take a lot longer to run than the previous steps. But, for the sake of time here, I'm again only running 1,000 iterations. So the parameters for spatial enhance are very similar to the earlier clustering analysis that we have up here. That there's a few differences. So the differences here are the jitter prior term and the jitter scale term. And so these are tuning parameters that help us, I guess, tune how the enhancement works, which I skipped this in my technical talk today because we didn't really go into a lot of the details here. But basically, our algorithm works by jittering or nudging the expression values in the spots into a higher resolution. And so these are some tuning parameters for that. The jitter scale is a parameter that we want to choose so that on average about 30% of your cells of your spots get jittered every single time. And we can check that in this way down here. So here I'm saving my MCMC chains. If I scroll down to the bottom, we're able to actually access these MCMC chains. So here I'm accessing a parameter called, called Y change. So this estimates the proportion of iterations that were successfully jittered. And we want this value to be approximately 30 to 40%. And so we see here, here are the first couple iterations. So this looks like it's only at about 20%. So perhaps if you run it for a little longer, you might see if this value changes and tune it in an appropriate way so that your values converge to about 30 or 40%. But yeah, the details for this are in our vignette. Okay, so once we have the enhanced clusters, we can again plot these enhanced clusters. And so comparing this to the previous image, there's still not too much enhancement going on here because we're only running it for 1,000 iterations. But if you run it for a longer period of time, then you'll get a result that looks more similar to what I'm showing in the slides over here. Okay, So here I'm showing enhanced clusters, but we're also able to get the enhanced expression of individual genes. So let's say that we want to enhance the expression of 4 marker genes. So here we have PMEL, CD2, CD19, and COL1A1. So these are markers for melanoma, the cancer, T cells, B cells, and fibroblasts. So we can use the enhance features function to do that. So there's a few options that I want to point out in the enhanced features function. So we have 2 different methods really for enhancing the expression of individual genes. So we do this by predicting the enhanced expression based on the enhanced principal components. So the methods are either to use XGBoost or linear regression. So the linear model basically does just a transformation back from the PC space to the gene expression space. And then XGBoost is a machine learning method for predicting. So in this case, XGBoost also has a tuning feature called N rounds that estimates the best number of boosting iterations. So by setting this into equal to 0, then we can automatically tune this. But if you're running enhanced features on a big data set, or if you want to enhance a lot of genes, it might be faster to just use linear regression or to set a fixed value for the number of boosting iterations here. So this actually finished pretty quickly because I have a pretty small data set, as well as only just a few markers over here. And so what happens now is that in the log counts for these 4 genes, I'll have the enhanced expression results. And so we can look at that right here. Here is the first couple of columns and the 4 genes that we showed previously. So these are the enhanced expression values. And we're also able to visualize these enhanced gene expressions using the feature plot function. So this is what the enhanced expression looks like here. And then just for reference, we can also look at what it looks like before the resolution enhancement. And so it looks like this over here. Okay, and again, for those of you who are more interested in the statistical side of things, to make sure that your MCMC chains are converging and things like that, then we can use the MCMC chain function to retrieve your chains and check for things like convergence. Okay, so this is the end of our first getting started tutorial, and then if anyone has questions, feel free to stop me at any time or put your questions in chat. I'll be moving on to our second analysis, which is a joint clustering analysis, actually. So I guess what a lot of users, recently have been asking us, can you do a spatial clustering analysis where you have multiple samples? And so I think we decided to work on that problem and see if we can come up with a solution to do a joint clustering analysis that takes into account the spatial nature of the data. And so we'll again actually use the dorsolateral prefrontal cortex data that I showed previously. And so here I'm going to do a joint clustering of 4 samples. So 151673 through 76. These are 4 serial sections from a single donor individual. So I won't be copying over the code anymore. So we'll just follow along in the vignette. But basically, first I'm loading in the 4 data sets using the getRDS function. So these 4 data sets are already preloaded into our package. And then I'm just plotting here the ground truth labels for these 4 samples. And so just from the image, it seems like there's a good chance that if we do some type of joint cluster analysis, we'll be able to share some information across different samples, and that might give us better results. And so there's a few things that need to be done to pre-process the data before we can actually do the clustering. So first is these data sets we've already pre-calculated which are the HVGs in order to do PCA. But in this case, since we're doing a joint cluster analysis, these need to be recalculated. So I'm setting these to null, and then combining all my individual single cell experiments into one big combined experiment that includes all four of my samples. And so once I have that, we can again use spatial preprocess to log normalize and do PCA on our data set. So when we do an analysis with multiple samples, it's always a good idea to check to see if there's a batch effect or some technical differences between your different samples. And in this case, from this UMAP, we can see that there is a pretty noticeable batch effect between the different samples. So since these 4 are replicates of one another, you would expect them to be, basically have the same structure, or for there to not be any noticeable structure between the different samples. But we can see here that clearly, some of these are, the green one especially, is separating out from the others. So before doing any type of joint clustering, it will be a good idea to first do a batch correction on this data set. So to do that, we use the Harmony package, which is, it's been shown previously to be pretty good at removing technical biases between samples. And it conveniently uses the single cell experiment format, so we can run it directly on our single cell experiment. So once we have harmonized the data, the results look like this. So now, after running Harmony, there is no clear batch effect anymore between the different samples. And so at this point, the data, the expression data is ready for clustering, but we still need to make sure that the spatial information is also set properly. So the row and the column information that determines your spatial location is not set uniquely for each sample. So in this case, I'm just adding some values to the row and the column values of the additional samples. So using 151673 as a base, I'm shifting over 74 to the right, 75 down, and 76 to this location here. So by doing this, we're now able to make sure that points from 2 different samples are not being counted as neighbors. So in this case, we're sharing information in terms of the clusters, but not in terms of spatial information between samples, because really, the actual physical distance between these replicates is a lot bigger than the physical distance between adjacent spots within each sample. Okay, so all of these pre-processing steps are done in terms of the batch correction and making sure the spatial locations are set properly. So at this point, we can run spatial cluster on the data set to perform a joint clustering analysis of all 4 samples. And so our results look like this over here. And so we see that there's a pretty nice layer structure for all of our samples, and the results look pretty consistent across the samples as well, which is a promising sign. we can calculate the adjusted RAND indices for these 4 samples. And so we get somewhere between 0.55 and 0.6 for all of these samples. And this is a pretty substantial improvement over our previous results, which were based on single sample clustering. So in this case, there was a lot more inconsistency between the 4 replicates. And once we do the data harmonization and the joint clustering, we are able to get a better clustering result across the whole data set. Okay, so I think I'm at the end of my allotted time for this package demo, and I think we can take questions now. Thanks.
All right, thank you, Edward, for the nice presentation. And we have one question. So I will, first question is from Saket Chudari, and he says, I will bring it on stage. Great work. How bad does naive jittering approach perform as compared to your approach? One naive approach would be to do partition on the observed expression simply based on the, I believe, nearest neighborhood distance, for example.
So I'm not super sure how a naive jittering approach would work. Basically, what our method is doing is we're jittering based on your spatial neighbors. So you're expected to get your expression to be closer to that of your neighbors. And it also takes into account your cluster label. So you're more likely to be jittering towards your cluster mean rather than in the opposite direction. So yeah, so I guess I'm not super sure what a naive jittering approach would be, because basically we're using the spatial information here to guide the direction of the jittering to in a way that it does smoothing while also doing a resolution enhancement. And the motivation or the inspiration for this is kind of drawn from some I guess like machine learning or computer vision ideas for how we for how we enhance resolution of images.
Right, Leo asked, and I will bring it on stage as well. If you are using multiple samples, would you increase the length of your Markov chain or use about the same?
So I think in our results, we saw that using the same number of iterations in the Markov chain worked pretty well. I haven't thoroughly tested this for convergence and like robustness over a bunch of samples, but it seems like just using the same defaults that we had previously worked pretty well on the prefrontal cortex data using multiple samples. The downside here is, of course, that if we have more samples, the joint cluster analysis takes longer than a single sample analysis.
All right, and another question from Marcus, naive practical question. For using the multi-sample BayesSpace enhancement in Syrava for workflow, I can do SC transform, Syrah batch effect correction, and do the same offsetting of coordinate tricks and run regular BayesSpace from here. That's a question.
Yeah. I think you definitely should be able to use Seurat for most of your processing steps, and then just use BayesSpace for the clustering part. Basically, there's I think Seurat and single cell experiment have some functions that allow you to interchange between the two data formats. But there's a few columns that don't get put into the right place. But if you fix those issues, then you should be able to use Seurat for most of your workflow, and then BayesSpace for just the clustering. And actually, one of our GitHub issues was an individual with this same problem. So yeah, if you have problems with that, just look through our GitHub issues and you'll probably find the answer or find a way to get around it.
All right, that's all our questions, and we are at the end of our time. Thank you, Edward, and thank you everyone who joined. That ends the first day of Bioconductor. Thank you again, and see you tomorrow.
Thanks. Bye, everyone.